sickle cell anemia. The example of autosomal is Huntington disease, or we call it Huntington's disease. And then we have the autosomal recessive, which is the examples are cystic fibrosis, and then we have sickle cell anemia. Then the second type, broad type of hereditary disorder is known as chromosomal defect, which involves uh, changes in the number or structure of the chromosomes. For example, Down syndrome is caused by an extra copy of chromosome 21. Another example, the Turner syndrome results from a missing X chromosome in females. Missing X chromosomes in females. Then you have another one, mitochondrial disorders, which results from mutations in the DNA of mitochondria. These are inherited exclusively from the mother, okay? From the mother. Now, so we have the single gene defect, we have the chromosomal defect, and then we have the microsomal disorders. Now, the symptoms of hereditary disorder. Symptoms vary widely depending on the specific disorder, but can include the following: behavioral changes, breathing problems, cognitive deficits, developmental delays, eating and digestive issues, limb and facial anomalies, movement disorders, neurological issues, poor growth or short stature, vision or hearing loss. Now, example of common hereditary disorders. We mentioned some of them: cystic fibrosis, Down syndrome, sickle cell anemia, Huntington's disease, Duchenne muscular dystrophy, Fragile X syndrome, thalassemia. Okay, those are common hereditary disorders. Of course, we know about cystic fibrosis, Down syndrome, sickle cell anemia, and then we have thalassemia. Those ones are we are quite familiar with them. And then that's all for now on hereditary disorders. So move swiftly to systemic lupus erythematosus, SLE. SLE. Now, SLE. Systemic and disseminated lupus erythematosus are the same autoimmune diseases. So if you are saying systemic LE or disseminated LE, you mean the same autoimmune disease, which is the systemic lupus erythematosus, which is SLE, in which the body's immune system attacks healthy tissue in multiple organ systems. It causes widespread inflammation and can affect the skin, joints, kidneys, brain, and other organs. Common symptoms include fatigue, joint pain, and skin rashes, but the disease is highly variable and can range from mild to severe. I take that again. The symptoms include fatigue, joint pain, and skin rashes, but the disease is highly variable and can range from mild to severe. What is SLE? It's an autoimmune disease. The immune system mistakenly attacks the body's own healthy cells and tissues. And it's also a multi-system disease, which can affect almost any organ system, including the skin, the joints, the kidneys, the brain, lungs, heart, and blood cells. And then it is highly variable. Symptoms vary significantly from person to person and can come and go. That is what we call the flare and remit. Okay? It's an autoimmune disease, it's a multi-system disease, and it is highly variable. Symptoms: you have fatigue and fever, unexplained tiredness, and low-grade fever are common. The skin: you have a butterfly rash across the face, especially on the cheeks and bridge of the nose. In a classic sign, it's a classic sign, rather, it's a classic sign. That is, a butterfly rash across the face, especially on the cheeks and the bridge of the nose, is a classic sign. Other rashes like viscoid, we call it raised scaly patches, or sun-sensitive ones can also occur. All right. Now you have joints. I'm still talking about the common symptoms in joint. You have joint pain and swelling. which are very common. Then you have internal organs. Inflammation can affect organs such as the kidneys, which we call lupus nephritis, and then the lungs, the heart, and brain. Other signs: headaches, hair loss, mouth sores, Raynaud's phenomenon, which is the finger and toes turning pale or blue in response to cold. It can also occur. Other signs: you have headaches, hair loss, mouth sores, and Raynaud's phenomenon, in which the fingers and toes turn blue or pale in response to cold. This can also occur. Then you have causes and risk factors. The first one is genetics. There is a genetic component to SLE, though the specific genes are not fully understood. Specific genes are not fully understood. Now, environmental factors. Environmental factors triggers, the environmental factors triggers, or the environment triggers, um, environment triggers are thought to be required to develop the disease in genetically susceptible individuals, which means that the environment, there are some things in the environment that can trigger the disease to develop in genetically susceptible individuals. We also have the hormones. The hormones. The disease is much more common in women, particularly those of childbearing age. Now we have the laboratory investigation. This is our duty. Complete blood counts with differentials. We have anti-nuclear antibody, ANA. They do chest X-ray, serum creatinine. You have urinalysis. We have anti-nuclear antibody, okay, panel. And then we have complement components to check the C3 and the C4. We also have the antibodies to double-stranded DNA. In the lab, sometimes you do what we call the early prep. In the hematology lab in USAID, we used to do it some times ago. The early prep, which takes close to 3 to 4, 5 hours to do, after which the slide, we examine the slide, and then we're able to identify through the help of a map to identify the the uh, the what do I call it now? The cells, yeah, the cells that are involved, the cells that are involved. All right, but we can use the map, the atlas of the atlas to be able to, map is atlas to be able to identify those cells, comparing them with the normal cells. And then you can also do what we call the C-reactive protein. And then we can do we have Coombs tests, the cryoglobulins, ESR, kidney function blood tests, liver function tests, and then you have the rheumatoid factor, and then the antiphospholipid antibodies, and then the lupus anticoagulant test that I talked about, the early prep. You can also do the lupus anticoagulant, which is like a coagulation-based test, to be able to identify if the patient has SLE. All right. You have the complications. Now you have lupus nephritis that affects the kidney. We mentioned it earlier on. Some people with SLE have abnormal immune deposits in the kidney cells. This leads to a condition called lupus nephritis. People with this problem may develop kidney failure. They may need dialysis or a kidney transplant. A kidney biopsy is done to detect the extent of damage to the kidney and to help guide treatment. If active nephritis is present, treatment with immunosuppressive medicines, including high doses of corticosteroids, along with either cyclophosphamide, or mycophenolates are needed. Okay, other parts of the body. Now, the SLE can cause damage in many different parts of the body, including the blood clots in arteries or veins of the legs, lungs, brain, or intestines. Then you have destruction of red blood cells, we call hemolysis, or anemia of long term, called chronic disease. Now, fluid around the heart, pericarditis, or inflammation of the heart, myocarditis or endocarditis. Fluid around the lungs, pleuritis, and damage to lung tissue. We have pregnancy problems, including miscarriage. Then we have stroke. We have bowel damage with abdominal pain and obstruction. Okay, so those are complications that affect what the SLE causes that affect the parts of the body that we have talked about now, and it's a whole lot of issue for SLE. And if from experience with experienced medical personnel with some of these investigations, they can easily detect, you know, um, or the onset of SLE, especially you have the rheumatoid factor and then the CRP, C-reactive protein. Let's continue. We have the inflammation of the intestines. We have the severe low blood platelet count called thrombocytopenia. Platelets are needed to stop any bleeding. So if it is low, there's a problem. Inflammation of the blood vessels. Inflammation of the blood vessels. Now let's talk briefly. Let's look briefly at SLE and pregnancy. You talked about it can cause. Uh, let me see now. I said it can cause. When you talk about the hormones, the disease is common in women, particularly those of childbearing age. So it can cause miscarriage. Can cause miscarriage. So let's look at the SLE and pregnancy. Both SLE and some of the medicine used for SLE can harm an unborn child. Of course, talk to your provider before you become pregnant. That's what advice we give to someone who needs help. If that person becomes pregnant, you find a provider who is experienced with SLE and pregnancy, because it can adversely affect the pregnancy and causes termination of uh, abrupt termination of the pregnancy. All right. So quickly, in that wise, again, let's look at rheumatoid arthritis. Rheumatoid arthritis, we've done SLE, we've done hereditary disorders briefly. We have looked at SLE briefly. Let's look at rheumatoid arthritis also briefly. So I think that we need in this 501 very briefly. Okay. Rheumatoid arthritis, or we call it RA, is a chronic. When you say something is chronic, that means it's ongoing autoimmune disease that causes pain, swelling, and stiffness in the lining of the joints, which we call synovium. It most commonly affects the joints in the fingers, hands, wrists, knees, ankles, feet, and toes. RA usually occurs in the same joint on both sides of the body, which makes it different from some other types of arthritis. This one is called rheumatoid. Please don't let us forget that rheumatoid. That's why I said rheumatoid arthritis usually occurs in the same joint on both sides of the body, which makes it different from other types of arthritis. There are so many types of arthritis. Uncontrolled inflammation damages the cartilage, which normally acts as a shock absorber in the joint. In time, this can deform the joint. Eventually, the bone becomes itself eroded, okay? And this can lead to the fusion of the joints. So that's just the little mechanism that occur. Uncontrolled inflammation damages the cartilage, which normally acts as a shock absorber. That means the cartilage acts as a shock absorber to the joints. With time, this can deform the joints. Okay, it can deform the joint because there's no more shock absorber. Eventually, that bone becomes eroded, and then this can lead to the fusion of the joints. Specific cells in the immune system, that's the body's infection fighting system, aids this process. These substances are produced in the joint, but also circulate and can cause symptoms throughout the body. In addition to affecting the joints, rheumatoid arthritis sometimes affects other parts of the body, including the skin, the eyes, the mouth, the lungs, and the heart. Okay? The skin, the eyes, the mouth, the lungs, and the heart. Four stages of RA. The first stage. In the early stage of RA, there is inflammation in the tissue around the joints. You know, they have some kind of joint pain and stiffness. If the provider orders X-ray, what we mean by provider is, of course, the clinician. They won't see destructive changes in the bones. If they just decide to do X-ray, they won't see any changes whatsoever in the bones. The first stage, there is inflammation, okay, and then the tissue around the joints, and then you start having some joint pains, and then some stiffness around the joints, okay. If you do X-ray, you may not see anything. The second stage, the inflammation has begun now to damage the cartilage in the joints. Then you might notice stiffness and a decreased range of motion in that area. Okay, the inflammation has begun to damage the cartilage in the joints. Okay, and then the stiffness will continue, and then there will be less motion in that area. Stage 3, the inflammation will become so severe that it damages the bones. The person will continue to have more pain, stiffness, and even less range of motion than in stage 2. You may need to start to see physical changes. The physical changes will start to show. The person will start to exhibit those changes physically. Okay. In stage 4, that is the final stage. The inflammation stops, but this joint keeps getting worse. You have severe pain, swelling, stiffness, and total loss of mobility. Okay, that is the 4 stages. First stage, don't forget, some joint pain and stiffness. Second stage, more stiffness and then decreased range of motion. The third stage, you have extreme pain, more stiffness, and then the motion becomes less than in stage 2. And then in 4, you have the inflammation, the joint becomes worse, joint pain becomes worse, and then there's extreme pain, there's swelling, there's total stiffness, and then absolute loss of mobility. It may take many years to go through all the stages, but some people don't progress through all the stages. There's always an intervention. There's always an intervention, and then you'll not be able to get to all the stages. Then we have the development of RA. How does it develop? In what age, what time, and when? The RA usually starts to develop between the ages of 30 and 60. It wouldn't surprise you that some people, even between 30 and 40, will start complaining of joint pain, severe joint pain from the knee, things like that. And then you say, it's arthritis or that. Can you have arthritis at the age of 30 or 40? Why? What is causing it? Because we think that arthritis occur even in elderly when they have done a lot of work in their prime, and then when it is time for them to rest, they start having all the pain. The body starts to compensate, start asking questions. But you see, you start having such things, at tender age now. It starts to develop between 30 and 60, but anyone can develop the condition. In children and young adults, usually between 16 and 40, I just said that now, it's called young onset rheumatoid arthritis, YORA. In people who develop symptoms after they turn 60, it is called later onset rheumatoid arthritis, LORA. So you have YORA for young people, and then you have LORA for elderly ones. Main symptoms. Symptoms of rheumatoid arthritis may include the following: pain, swelling, we've said that, stiffness, tenderness in more than one joint. You have stiffness, especially in the morning or after sitting for a long period of time. You have pain and stiffness in the same joint on both sides of the body. You have fatigue, which is extreme tiredness. You have weakness, you have fever. RA affects everyone differently. In some people, symptoms develop over several years. In other people, rheumatoid symptoms progress rapidly. Many people have time with symptoms, we call it flare, and then time without any symptoms. That is remission. Sometimes it comes, sometimes it goes. And then you just feel, I don't have it anymore. And then later it just surfaces again. Then what are the early signs? Early signs of RA include tenderness or pain in the small joint, like the finger and toes, or then might notice pain in a larger joint, like a knee or shoulder. These early signs of RA are like an alarm clock set to vibrate. It might not always be enough to get your attention, But the early signs are important because the sooner they are diagnosed with RA, the sooner the treatment can begin so that we don't go through the 4 stages. Also, prompt treatment lowers the risk of developing permanent, painful joint damage. Complications. Without treatment, RA can lead to further complications, which include, but not limited to, the following: damaged or deformed joints, viral infections, dry eyes and dry mouth, carpal tunnel syndrome, osteoporosis, heart disease, interstitial lung disease, and then lymphoma. Complications again: damaged or deformed joints, viral infections, dry eyes or dry mouth, carpal tunnel syndrome, osteoporosis, heart disease, interstitial lung disease, and then lymphoma. Diagnosis and tests. To make an RA diagnosis, the healthcare provider may refer to a doctor who specializes in arthritis. We call them rheumatologists. Rheumatologists. They diagnose people with different RA based on the combination of several factors, and they'll do a physical exam and ask persons for medical history and symptoms. Now, rheumatologists can also order investigations that concerns us. The blood tests look for inflammation and blood proteins, which we call antibodies, that are signs of rheumatoid arthritis. This may include ESR, we said it the other time, CRP, and the rheumatoid factor antibodies. Then you have cyclic citrulinated peptide, CCP antibodies. CCP antibodies are the latest, mode of diagnosis of the uh, the the RA. However, it's not common. It's not common because it is extremely expensive and it has a lot of procedures. But it's becoming a little less complicated now in developed countries because it's easy to look at. Next, okay. So that is that about 501 for now. I don't know if we have any question before we continue. Like we have all said and agreed, our topic is yes. Mr. Akibile, Adebola, floor is yours, please.
Thank you very
Before
much,
we
sir. Good morning,
yeah,
sir. Thank you for all all you have been teaching us and your efforts. We really appreciate it. It's been very explicit and easy to really assimilate. I want to, I don't know if it's a question. In this case of lupus, SLE, I've seen
yeah,
a case, you mentioned the age range, and I could say, I
yeah,
think you said 30 and all of that, from 30
yeah,
years. But I've seen a case very close to me that had, of course, still battling with it from age, she started in age, she was in secondary school, I think JS2, 3. Although I learned it's very, very rare, but I've seen a case like that. She's still in it, actually, but at least we are managing her. A girl of age 13, they're about 13, 14. So I think
yeah.
maybe it's an aberration, I don't know, but it happens too. That's just the information I have. Thank you, sir.
Yes. It can be one out of many. It can be one out of many. So if you have a child that is between 10 and 15 and uh, has or is down with SL, it's possible. It's very, very possible. But at that age, it's easy to treat. It's very easy to treat, especially if you are able to get you know a provider that is specialized in that. Because it's not everyone that can do that, that can treat. People just open their AI and start checking for treatment and things like that. You have experienced people who be able to do that. But you see, it's not an aberration. It can happen. It can happen. It's possible. Very, very possible. All right. Very possible. Thank you for the comment. Any other one before I go on to talk about our CA? Okay. So please, I have not put it here on the slide, but if you have something to write on, please write right now, and then the class rep will paste it again. Okay, for 501, we are going to have 2 assignments. 501, we're going to have 2 assignments. Okay, and then you will submit the assignments as a CA to my mail. Okay, the class rep will also post the mail. So that will be all for 501. But let me just dictate it while she will get it posted. We are going to write a short note on the following. A, hemoglobin genotype. That's A. And B, hemoglobin phenotype. Genotype A and phenotype B. That's the first question. Then the second question. Second question. Okay. Yes, are we here? I hope you are listening. Okay.
Yes, sir.
Now, yeah, thank you. The second question is discuss blood in infancy, childhood, and pregnancy. Discuss blood in infancy, childhood, and pregnancy. Those are the 2 questions for the CA. Okay? You can start to submit as soon as you can lay your hands on it. And then it's something that is very simple, very, very simple. So those are the assignments for 501. Okay, so I'll give the class rep the assignment again, and then we'll do also the email. Okay, so all right. Let me move back to the 5. We are going to 503 immediately now. So just give me a few minutes while I go to the slides, please. Just give me a few minutes while I go to the slide. Okay. All right, so we're going to go to 503 quickly. 503 is blood transfusion science, and we're going to go as fast as we can, all right, and then conclude upper week, and then make sure that you're done, if we can, whatever, we still have class tomorrow, if it is possible, and finish. If not, we'll continue from where we stopped, and then we give ourselves some. So let's go to the first slide, which is the ABO blood group. All these ones we did in our 400 level. So, and we all know it. So there's no point wasting too much time. And then I'll just run through the slides. The discovery of ABO blood group over 100 years ago caused great excitement. Until then, all blood had been assumed to be same, and then often tragic consequences of blood transfusions were not understood. As our understanding of ABO group grew, not only did the world of blood transfusion become a great deal safer, but scientists could now study one of the first human characteristics proven to be inherited. A person's ABO blood type was used by lawyers in paternity suits, by police in forensic science, and by anthropologists in study of different populations. So you can use it to do anything. And then the ABO blood group antigens remained of prime importance in transition medicine. They are the most immunogenic of all the blood group antigens. The most common cause of death from a blood transfusion is a clerical error, in which an incompatible type of ABO is transfused. The ABO blood group antigens also appear to have been important throughout our evolution because the frequencies of different ABO blood types vary among different populations, suggesting that a particular blood type conferred a selection advantage, e.g., or for example, resistance against an infectious disease. However, despite their obvious clinical importance, the physiological functions of ABO blood group antigens remains a mystery. People with the common blood type O express neither the A nor the B antigen, and they are perfectly healthy. Numerous associations have been made between particular ABO phenotypes and an increased susceptibility to disease. For example, the ABO phenotype has been linked with stomach ulcers, more common in group O individuals, and gastric cancer, more common in group A individuals. Another observation is that individuals with blood type O tend to have lower levels of the von Willebrand factor, which is a protein involved in blood clotting. So these are possibilities. Okay. In the beginning of the 20th century, an Austrian scientist, Karl Landsteiner, noted that the RBCs of some individuals were agglutinated by the serum from other individuals. We all know this, but we have to say it. He made a note of the patterns of agglutination and showed that blood could be divided into groups. This marks the discovery of the first blood group system, ABO, and earned Landsteiner a Nobel Prize. Landsteiner explained that the reactions between the RBCs and the serum were related to the presence of markers, antigens, on the RBCs and antibodies in the serum. Agglutination occurred when the RBC antigens were bound by the antibodies in the serum. He called the antigens A and B, and depending upon which the antigen the RBC expressed, blood either belonged to the blood group A or blood group B. A third blood group contained RBCs that reacted as if they lacked the properties of A and B, and this group was later called O, after the German word own, and which means without. The following year, the fourth blood group AB was added to the ABO group system. These RBCs express both A and B antigens. These are things that we know, but we need to mention them. Okay, for us. In 1910, scientists proved that the RBC's antigens were inherited and that the A and B antigens were inherited co-dominantly over O. There was initially some confusion over how a person's blood type was determined, but the puzzle was solved in 1924 by Bernstein's three-allele model. The ABO blood group antigens are encoded by one genetic locus, the ABO locus, which has three alternative allelic forms, the A, B, and O. A child receives one of the three alleles from each parent, giving rise to 6 possible genotypes and 4 possible blood types, which is phenotypes. Now, the ABO phenotypes. The 4 basic ABO phenotypes are O, A, B, and AB. After it was found that blood group A, RBCs reacted differently to a particular antibody, later called anti-A1. The blood group was divided into 2 phenotypes, A1 and A2. RBCs with A1 phenotype react with anti-A1 and make up about 80% of blood type A. RBCs with A2 phenotype do not react with anti-A1 and they make up about 20% of blood type A. A1 red cells express about 5 times more A antigen than A2 red cells. But both types of red cells react with anti-A. And as far as transfusion purposes are concerned, the A1 and A2 blood groups are interchangeable. These are things that we know already. Now, ABO antibodies in the serum are formed naturally. Their production is stimulated when the ABO immune encounters the missing ABO blood group antigens in foods or in microorganisms. This happens at early age because the sugars that are identical to or very similar to the ABO blood groups antigen are found throughout nature. Now, ABO locus has three main allelic forms. We have said it earlier, the AB and the O. The A allele encodes a glycosyl transferase that produces the A antigen. Okay? That is the N-acetyl galactosamine in its immunodominant sugar. And the B allele encodes glycosyl transferase that creates the B antigen as a D-galactose, it's immunodominant sugar. Okay, why we are doing ABO and really talking more about ABO is because it's the most popular, and this is, what we use for our compatibility testing. All right. So we have two, the sugar. We have mentioned N-acetyl galactosamine, and then we have mentioned the D-galactose. Now, diseases associated with ABO blood group antigens. We had to, from where I extracted it, we had to use some references. You know, if you need the references, I can give it to you. But I just kept it for my own convenience. So you can ignore the 4, the 5. Those are the references that I used. Now, disease associated with ABO blood group antigens. No diseases are known to result from the lack of expression of ABO blood group antigens, but the susceptibility to a number of diseases has been linked with a person's ABO phenotype. Such correlations remain controversial and include the observation that the gastric cancer appears to be more common in blood group A individuals. Okay, the person that did the research is the one that I quoted there. And then whereas gastric or duodenal ulcers occur more often in group O individuals. A clear correlation has been established between the ABO phenotype and the level of two proteins involved in blood clotting. Now, factor 7, factor 7 in brackets, which is factor 8 and combination of factor 8 and von Willebrand factor, And okay, let me take that place again. Let me take the place here. A clear correlation has been established between the ABO phenotype and the level of two proteins involved in the blood clotting factors. We talked about the factor 7, we talked about factor 8, and then the von Willebrand factor. Blood group O individuals have about 25% less factor 8 and von Willebrand factor in their plasma. It is well established that low levels of factor 8 and von Willebrand factor are a cause of excess bleeding, and therefore it may also be the case that increased levels makes clotting more likely. Okay? Now, increasing the risk of both arterial, which is the ischemic heart disease, and venous, which is the thromboembolic disease problems. Indeed, the non-group O individuals have been shown to be at a increased risk of both arterial and venous disease. Now, what I just dictated to us now, are things that are associated. These are associated with the ABO blood group antigens, and these are things. The research that have been carried out by some individuals. That's why I quoted the numbers for my own referencing. These are people who have established, who have been able to establish this research. And even though it has not been fully authenticated, that this is really so, because we have not seen, if you have duodenal ulcer for people who are blood group O, so a lot of people who are blood group O, I'm not sure they have duodenal ulcer or any form of ulcer in that case, or people who are blood group A who have gastric cancer, you understand? So we have to check all those things. But these are work that people have done to show that, okay, there are some diseases that are really associated with ABO blood group antigens, and it's associated with some who are of different blood group status. Now, clinical significance of ABO antibodies. Now, ABO antibodies are of major clinical significance for two reasons. They are naturally occurring and are found universally, and they are highly reactive. And that's why we are studying them. Okay? Very, 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 reactive. And it is naturally occurring. Now, transfusion reaction. transition reaction. We've talked about compatibility testing. Adding the resource factor to it makes it more complicated when it comes to the compatibility testing. We'll still come to that, briefly. Transfusion reaction. The routine practice of blood typing and cross-matching blood products should prevent adverse transfusion reaction caused by ABO antibodies. However, clerical error can result in wrong blood being transfused to a patient, an error which can result in the death of a patient. Now, 7 and 8, let me just tell us quickly what they said, what they discovered. Now, they discovered that, which we also know, basically clerical error sometimes does not really, well, we do not really have clerical errors often happening nowadays. But from the study, from the research that they have done, people that have died through blood transfusion reaction, they discovered that majorly it's not even incompatibility, it is clerical error. It is clerical error that had caused the death of those patients. After the blood has been said to be very compatible from the cross-matching room and is set to be dispatched, that is where the problem is. And or when the sample is being received and there is a clerical the people at the clerical section making made some error. You understand? So that is what those people have done. And of course, people have done it again too in our climb here. And they've discovered that apart from ABO incompatibility, we have clerical error that is a major problem in compatibility. Now, if a recipient who has blood group O is transfused with non-group O RBCs, the naturally occurring anti-A and B in the recipient serum binds to their corresponding antigens on the transfused RBCs. These antibodies fix complement and cause rapid intravascular hemolysis, triggering an acute hemolytic transfusion reaction that can cause disseminated intravascular coagulation, shock, acute renal failure, and eventual death. Anti-A1 is a less significant cause of transfusion reaction and does not appear to fix complement. We talked about A1, A2 the other time. Now, hemolytic disease of the newborn. I'm not going to stress myself on this because somebody is doing that for what's that seminar. So I'm just going to paraphrase it and then move on from there. I'm going to paraphrase it and move on from there. Okay, we still have. Wait a minute. Let me see. Okay. Wait, before I come to HDN, which is very brief, let me just go to the other blood group systems. Let me just go to so that we will just follow a particular sequence. So let's go to the resource. First, let me just make an introduction. Beyond the well-known ABO system, which we have talked about, I've talked about ABO blood group system. You're a hematology scientist. We don't need to flog that. There are numerous other blood group systems, such as the resource, which is the second most important. I agree totally. And other systems like Duffy, the KEL, the KID, and Lutheran systems, totaling over 48 recognized blood group systems. These systems are defined by different antigens on the surface of red blood cells and can be relevant for blood transfusions as antibodies to these antigens can cause transfusion reactions. Now let's look at the resource. Resource is the second most common. So let's look at the resource. Resource system is the second most important blood group system after the ABO. Currently, resource system consists of 50 defined blood group antigens, out of which only 5 are important. RBC surface of an individual may or may not have a resource factor or immunogenic D antigen. Accordingly, the status is indicated as either resource positive, that D antigen is present, or resource negative, which means the D antigen is absent. In contrast to the ABO system, the anti-resource antibodies are normally not present in the blood of individuals with D negative RBCs, unless the circulatory system of these individuals has been exposed to the D positive RBCs. These immune antibodies are immunoglobulin, IgG in nature, and hence can cross the placenta. Prophylaxis is given against rhesus immunization using the anti-D Ig for pregnant rhesus-negative mothers who have given birth to rhesus-positive child. That is what we call the ROGAM that is given. Okay, that's rhesus in a nutshell. We don't need to flog all these things. Just understand it basically and just put it down in case you are asked. Even though we'll have revision exercise later, and then we'll be able to expose ourselves to what can be asked and things like that as usual. So we have MNS. MNS antigen system, first described by Landsteiner and Levine in 1927, is based on two genes rather, glycophorin A and glycophorin B. The blood group is under control of an autosomal locus or chromosome 4, and also under control of a pair of codominant alleles, LM and LN. Okay? Anti-M and anti-N antibodies are usually IgM types and rarely associated with transfusion reaction. As simple as ABC. Okay, simple, very simple. Lutheran. Lutheran system comprises of 4 pairs of allelic antigens representing 6 amino acid substitution in the Lutheran glycoprotein at chromosome 19. Antibodies against this blood group are rare and generally not considered clinically significant. Care. These erythrocyte antigens are the third most potent immunogenic antigen after ABO and rhesus. So after ABO and rhesus, the next is the KEL, okay? And are defined by an immune antibody called anti-K. It was first noticed in the serum of Mrs. Kelacher. Mrs. Kelacher. She reacted to the erythrocytes of a newborn infant, resulting in hemolytic reactions. Since then, 25 KEL antigens have been discovered. Anti-KEL antibody causes severe hemolytic disease of the fetus and newborn and hemolytic transfusion reactions, HTR. So anti-KEL or KEL system is also very important and it is very dangerous. It's dangerous. Okay, so let's know that ABO, resource, KEL, they come after one another like that. Then we have the Duffy. We're just looking at very few of them. Duffy antigen was first isolated in a patient called Duffy who had hemophilia. It is also known as FY glycoprotein and is present in the surface of RBCs. It's a non-specific receptor for several chemokines and acts as a receptor for human malaria parasite, the Plasmodium vivax. Antigens FYA and FYB on the Duffy glycoprotein can result in 4 possible phenotypes. We have namely FY, A plus B minus, FY, A plus B plus, FY, A minus B plus, and FY, A minus B minus. The antibodies are IgG subtypes and can cause hemolytic transfusion reaction. The next one is the KID. The KID antigen is known as the JK antigen and is a glycoprotein present on the membrane of RBCs and acts as a urea transporter in RBCs and renal endothelial cells. KID antibodies are rare but can cause severe transfusion reactions. These antigens are defined by reactions to an antibody designated as anti-JKA discovered by the serum of Mrs. Kidd, who delivered the baby with HDFN, that is hemolytic disease of newborn. Then the JKA was the first antigen to be discovered by the Kidd blood group system. Subsequently, 2 other antigens, JKB and JK3, were found. Okay, so let's go to HDN. It will make more sense, but it's very brief. So the person that is taking us HDN on seminar will give us a fuller picture in the seminar. Most cases of HTN that arise from an ABO compatibility require no treatment. Cases of severe hemolysis that require exchange transfusions are less common, and fetal eye drops is rare. HDN caused by ABO antibodies occurs almost exclusively in infants. Now, I must say that the HDN that I'm talking about now is the HDN that affects the ABO. We have the HDN that affects the resource. Okay, I'm sure the person that is going to will do justice to that. So let me just do this briefly for the ABO. Okay, now HTN caused by ABO antibodies occurs almost exclusively in infants of blood group A and B who are born to group O mothers. This is because the anti-A and anti-B formed in group O individuals tend to be of the IgG type and therefore can cross the placenta, whereas the anti-A, anti-B found in the serum of blood group B and A individuals, respectively, tends to be of the IgM type. Although uncommon, causes of HDN have been reported in infants born to mothers with blood group A2 and blood group B. HDN tends to be relatively mild in nature, mainly because fetal RBCs don't express adult levels of A and B antigens. However, the strength of fetal ABO blood group antigens can vary, and therefore the degree of hemolysis and hence the severity of HDN can be unpredictable. Early studies suggested that the race for a neonate was a risk factor for developing ABO-HDN. However, later studies showed that the prevalence of disease that required treatment did not suffer significantly among some Blacks and some Caucasian individuals. What that is simply telling us is that, you see, the ABO-HDN is rare, in some people. But the major HTN that is responsible for fatality is a resource, and that's a resource HTN that I'm sure that the people that are doing seminar are going to expose us to, and that's what academic discourse is all about. So, but that's the ABO HTN. You have two types. You have the ABO ADN, and then we have the resource HTN. All right. So, okay, let me stop there for today. We'll continue tomorrow from here, hemolysin titration. We continue from there tomorrow and see how far we can go. Do we have any question? You can unmute your mic if you want to say anything. Have any questions? Then we can make some announcements. Okay. So we started on the 503 now. The 503, 503, yes. Started on 503. So we'll continue from there tomorrow. And we really dealt with ABO, and then we have the other blood groups, and then the ABO HDL for this morning. So any question, please? Is the class rep here? Is the class rep online?
I don't think so.
Okay.
She's not. She's
She's
not.
not. She's not.
Okay, no problem. Do you have any question at all? For those who are in my group for supervision or for supervisory roles and all that, please, we'll go on our page later and then discuss one or two things so that we can move. And I know Mr. Adeyemi talked about that again the other time, so please, let's move fast in hematology. We'll be having deadlines. We said, by the grace of God, we'll finish our lectures by the end of this November, which we are moving to do. And then see by the side. We prepare our seminar, and then we'll continue with our project. By the end of January, February, we should be done, and then be ready to continue with the second semester and move with the speed of light. We do not have time. We are all very busy, and you know that very soon, once it is time, we'll do our revision, and then we'll know what to read and all that, so that I'm not stressed. It is not stressing at all. It's not going to be stressing at all. So let's just put our mind at rest. Hematology is going to be swift and straight. Okay, and God will help us so that we can move with the deadlines that we have. We will not, be stressed at the later age and be doing fire brigade when the department or the dean start to pursue or submit this, submit that. Hematology should be 100% to just submit and be at home and just be waiting for others. I hope I'm clear so that we can move together. We can all be on the same page. I know some of us might not be as fast as possible. That's why we are creating the time. We are creating the time so that we can all move together. Okay? And all move together in that speed. All right. So if you don't have any other question or any comment, do you have any question aside what I taught today? Is there anything you want us to talk about or clarify in hematology as we progress? Okay. Okay, if there is none, I want to appreciate everyone who were able to make this morning's lecture. And it's not been easy, but we have to do it. I want to thank you so much. We'll meet again 6 p.m. tomorrow. I think from what the class rep posted, this is going to be our Zoom link until further notice. I think maybe that's the way I interpret it. Maybe this is going to be the only Zoom link that we'll be using for now until it's said otherwise. All right. So thank you very much, everyone. I want to appreciate us so we can go back to doing whatever we are doing for the weekend. So have a wonderful and beautiful, beautiful day. So we'll talk tomorrow
Thank you,
again in the
sir.
evening.
Thank you, sir.
God bless you, sir.
Thank you, sir. God bless
Thank you,
you.
sir.
All right,
Thank you.
sis. All right, Mas. God bless you. All right.
Have a nice weekend, sir.
You too. You too. Yes, sir. Yes, sir.